part of the semester, we were really focusing on individual neurons, right? We talked about communication within the neuron in terms of resting membrane potential and then the action potential traveling down the length of the axon to send the message within the same cell. And then we talked about communication between neurons in terms of synaptic plasticity. And we were really taking a close look at what exactly happens when the action potential reaches the terminal, how neurotransmitter is released, and all of the things that can happen to that neurotransmitter once it has been released into the synapse. Now we're going to take a step back and look a little bit bigger picture, okay? So the first thing we're going to start with is simple neural networks. This information comes from Neuroscience Online. We're going to talk about neurons as either being an excitatory neuron or an inhibitory neuron. 75% of the synapses in our central nervous system use the excitatory amino acid neurotransmitter glutamate. These are the excitatory synapses. 75% of CNS synapses are excitatory. What does it mean to be an excitatory neuron? Other than it releases glutamate. Releases amino acid neurotransmitter glutamate.
I think excitatory neurons, they have action potentials.
All neurons will have action potentials, well, most.
It reaches threshold.
No. What does glutamate do to the postsynaptic cell?
It's more like there's a cause and action potential on the postsynaptic cell.
Why?
Because it releases a neurotransmitter that allows EPSP to put cations in.
So glutamate is the neurotransmitter, and when it binds to its postsynaptic receptors, it will typically cause an EPSP. So an excitatory neuron releases glutamate. Glutamate causes an EPSP on the postsynaptic membrane. And as Jeanette was saying, that increases the probability that the postsynaptic cell will reach threshold. %20 of the synapses in our central nervous system use the amino acid neurotransmitter GABA or glycine. And these are inhibitory neurotransmitters. I'll come back to you in a second, that's okay. What does it mean to be an inhibitory neurotransmitter or to be an inhibitory neuron? Sam?
The GABA or glycine binds with the metabotropic receptors and, or wait, I think I'm getting that wrong. Is it ionotropic or metabotropic? Because I know it causes an IPSP.
It causes an IPSP, right. And the most direct way to do that would be which type of receptor? The ionotropic is the most direct way to cause an IPSP. So inhibitory neurons release the amino acid neurotransmitter GABA, or in the spinal cord, glycine. Remember, central nervous system encompasses the brain and the spinal cord. Okay, so inhibitory neurons release the amino acid neurotransmitter GABA or glycine and cause an IPSP on the postsynaptic cell. And remember, EPSPs and IPSPs, postsynaptic potentials, are local graded potentials. This is a change in memory potential on the postsynaptic cell right at the location of the synapse. And those charges can only travel so far, there's no regeneration. Okay? So we've got 75% of synapses use glutamate, 20% use an inhibitory neurotransmitter. What's left? To get to 100%. 5%. 5% of the CNS synapses use a neurotransmitter that is not an amino acid, not from that category. So that means acetylcholine, serotonin, dopamine, norepinephrine, all of those combined make up about 5% of the CNS synapses. Now, how many synapses do you suppose exist in our central nervous system? A lot. And what is 5% of a lot? A lot, right? So 5% of the synapses is still a lot of synapses. I don't want you to look at these numbers and think, oh, monoamines aren't important. That's not the case. They're still absolutely important. It's just that most of our synapses are either excitatory or inhibitory. And then we have this other 5% that maybe are modulatory. Now let's talk about how we can put excitatory and inhibitory neurons together in order to achieve a particular function. To start, we'll talk about these basic neural networks. This information, again, comes from Neuroscience Online. Let's start with feed-forward excitation. So I'm using this color code here, right? Excitatory neurons in green. Is anybody red-green colorblind? No? Are you? Can you see it still? Can you tell the difference? I can
yeah,
do.
If I sit here, it should be fine.
I'll do a square shape for the inhibitory neuron. Okay. All right. So the excitatory neuron, cell 1, when it fires an action potential, what neurotransmitter is it going to release? Glutamate. Okay. And then the postsynaptic cell, cell 2, glutamate will bind to receptors on the postsynaptic membrane, and it will cause what? Glutamate can't cause an action potential. What can glutamate do? It can bind receptors and cause an EPSP. Feed-forward excitation is simply combining a couple of excitatory neurons in a row. Cell 1 says, hey, cell 2, I have something interesting to tell you, and cell 2 passes that message along. Okay? Now, feed-forward inhibition. How do you suppose it's different from feed-forward excitation? Well, one of the neurons in there has to use GABA or glycine, right? So here we have cell 1. If cell 1 fires an action potential, what neurotransmitter will it release? If cell 1 fires an action potential, it's still green. Glutamate. Okay. Now we have our inhibitory neuron, cell 2, which is a square. So if cell 1 fires an action potential and releases glutamate, what happens on the postsynaptic membrane at cell 2? An EPSP. Let's say that EPSP is sufficient to reach threshold. Then what happens in cell 2? Action potentials. Cell 2 fires action potentials. What neurotransmitter does it release? Let's say we're in the brain.
Glycine.
In the brain, it's GABA. In the spinal cord, it's glycine. GABA is going to bind to receptors on the postsynaptic membrane of cell 3. And what will happen to the postsynaptic membrane of cell 3? An iPSP. Is cell 3 more or less likely to fire action potentials? Less. Okay? Now we have a couple of feedback circuits. Whenever we're talking about feedback circuits, it always feeds back to the actual initial neuron, to cell 1. Feedback inhibition, we've got cell 1. Cell 1 fires action potentials. What neurotransmitter will it release? Glutamate. what effect will that have on cell 2? EPSP. Let's say that's sufficient to reach threshold. What neurotransmitter is cell 2 gonna release? And what effect will that glutamate have on cell 3? EPSP. Let's say that's sufficient for cell 3 to reach threshold. Cell 3 fires an action potential. What neurotransmitter does it release onto cell 1?
Is this feedback inhibition or?
This is feedback inhibition.
Okay.
And then what happens to membrane potential of cell 1 at the postsynaptic membrane? An IPSP.